right at noon. We're just trying to give everybody a good chance to join us. We'll be starting right at noon.
Recording in progress.
Good afternoon and good morning, everybody. We're going to give it just a couple of more minutes. We're expecting a very, very big attendee roster today, so we want to give everybody a good chance to join us. So we're going to give it just another minute or two. We're expecting over about 500 people, so we're excited to give this presentation today. Dean, can you let me know how many we have so far?
Yep, 141.
All right, we're going to give it another minute or two. Make sure everybody's with us. Gideon, are we over 200 yet?
We are close. We're at 175.
All right, good afternoon and welcome. Good morning to the folks in the Western time zones, and good afternoon to everybody here on the East Coast. My name is Isaac Miller, and I am with the FDA's Office of Scientific Professional Development. Thank you for joining us for today's FDA Grand Round, sponsored by the FDA's Office of the Chief Scientist. Our lecture for today will be provided by Dr. Elena Skizakova. Before we begin today's Grand Rounds, I have a few CE and class instructions to read. Speakers are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. Disclose unapproved use. CE speakers are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations on the information that is presented. Disclosures. Today's speaker, the FDA faculty, planning committee, and CE consultation and accreditation team have nothing to disclose. Continuing education. This activity is approved for 1 CME, CPE, and CNE contact hours. Continuing education credit will be available for those who attend via Zoom. Pre-registration via Zoom is required. Requirements for continuing education. Attend the activity verified by Zoom registration. Physicians, pharmacists, nurses, and those claiming non-physician CME must attest to their attendance and complete the final activity evaluation via the CE portal. We encourage all participants to complete the post-activity survey as it provides valuable feedback which may affect future activity offerings. Instructions on how to complete the survey, claim CE credit, and or print a certificate will be emailed to you within 24 hours of this session. The claiming code is on the screen there. I'll give folks a moment to write it down if they want to go ahead and get a head jump on claiming directly after the class. If not, it will be emailed to you sometime tomorrow. Zoom. Closed captioning is provided for today's seminar. If you do not see the CC, press the CC button at the bottom of the screen. There will be a Q&A session at the conclusion of today's talk. Please put all questions in the Q&A pod at the bottom of the screen. This webinar is being recorded. The recording will be posted on the website within 5 days post-event. All right. Today's speaker, Elena Sizikova is a staff fellow in the Office of Science and Engineering Laboratory Center for Devices and Radiological Health in the Food and Drug Administration located in Silver Spring, Maryland. Her work addresses research problems at the intersection of artificial intelligence, medical imaging, and computer vision, focusing on the use of synthetic data in regulatory science. The title of today's presentation is Synthetic Data for Medical Imaging AI. All right, time for our esteemed speaker coming from the White Oak campus, FDA, Silver Spring, Maryland. Elena, is all yours.
Thank you so much for the introduction, and thank you for the opportunity to discuss my work. Let me share my screen. Are you able to see my screen now?
Yes, ma'am.
Great, thank you. My name is Elena, and I'm a senior staff fellow in the Division of Imaging Diagnostics and Software Reliability at U.S. Food and Drug Administration. And it's an honor to present our work today to you. So first, I'd like to start with some slides that discuss what I'd like to discuss today. So I want to talk about different classes of techniques for generating synthetic medical imaging data and summarize their strengths and weaknesses. And I want to also discuss how synthetic data generation for some practical applications such as breast and skin imaging can be effectively used for various stages of medical AI development. Oops. So, what is DITSER? DITSER is a division within the Office of Science and Engineering Labs, also, which is the research arm of the Center for Devices and Radiological Health within FDA. At DITSER, we conduct regulatory reviews of AI-enabled medical devices and develop regulatory science tools to accelerate and facilitate a review of the types of devices that we see daily. In this chart, you can see where regulatory science tools and publications are part of the regulatory tools evolution. And RSTs are a new way to de-risk technology and development by providing practical solutions for evaluating devices. RSTs do not replace other regulatory tools that we employ during the FDA review process. But one notable key benefit of RSTs is that it takes less than a year to get them out of our hands and into the hands of the stakeholders. On the other hand, the development of guidance documents is typically in the ballpark of 3 to 6 years, while standards take even longer and in the range of 5 to 12 years. Here's a snapshot of the RST catalog, which is already 3 years old as of today. You can access this catalog online. It's freely available. And early data suggests that the product catalog has approximately tripled the use of the tools that are part of it. We're adding new tools at the rate of 50 a year and looking to significantly expand the capacity. And I want to emphasize that a lot of these tools, there are links to the data, the code, links to GitHub, and you can sort of interact and open issues and really really be able to see how you can use these tools in practice and further and further. So today I want to focus on one of the major gaps and challenges facing medical imaging AI, which is the limited and labeled data sets. In this slide, you can find some data about the number of device submissions to the agency that contain an AI component. And this is public data across many product areas. And look at what happened in the last few years. There has been a dramatic increase dominated by radiological applications, but permitting to other device areas. And we expect this trend to continue, and we don't see any indications of slowdown. So what is common across all of these devices? Medical devices need to be evaluated on representative populations, and particularly AI algorithms require access to large training and testing data sets that would have enough variability to demonstrate the safety and effectiveness in the intended use populations. On the other hand, real patient data sets that are commonly used to within development are often limited to a number of factors. They're often smaller in size and constrained in variability. They may not represent rare or life-critical cases. They may be unavailable to privacy regulations, costs, or risks of acquisition, and as a result, they may be biased. And even when this data is available, they may not have labels of the type of information that may be needed to develop a particular device. So data availability is a huge issue in medical device development. And one of the potential solutions to the data availability is synthetic data. So as defined by the artificial intelligence glossary, synthetic data is artificial data that is intended to mimic the properties and relationships seen in real patient data. Synthetic data are examples that have been partially or fully generated using computational techniques rather than acquired from a human subject by a physical system. So some initial comparisons of patient and synthetic data you can see here. So patient data is human data, while synthetic data is an approximation. So that's a negative for synthetic data. On the other hand, patient data is more burdensome to collect, while synthetic data often doesn't need, can be collected in much easier fashion and can be simulated. Patient data may contain limited samples, while synthetic data if you have access to a good compute system, you can generate unlimited samples. Patient data may have limited patient variability due to the challenges in acquisition, but in synthetic data, you can tune the variability that you like. Patient data is often more narrow in scope, while synthetic data is adjustable. In patient data, some of the reference standard or truth may be uncertain, while in synthetic data, often it's easier to obtain the labels. Also, synthetic data, it may reduce the risk of data collection, so there's no inherent risk, for example, radiation risk when imaging a patient. Patient data may also pose privacy concerns for data sharing, while synthetic data, again, depending on how it is generated, may have less privacy concerns for further data sharing. And finally, patient data for all of these factors individually or together may be biased, and synthetic data, depending again on the way it is generated, we may have more control over the biases that we know, and we can potentially be able to address them. So as you could see, synthetic data poses a number of advantages or potential advantages to patient data. And the question is, can synthetic data partially or fully replace real data? For AI and commonly classification, detection, or segmentation algorithms, can we simulate enough synthetic data for AI development? And in this slide, I show some sample synthetic mammography images that we developed in-house to support some of the testing to evaluate this claim. I will present the MSIM data set in a later slide. So here I want to show an image of a breast cancer lesion, and I want to ask you which image contains a synthetic image and which one is not, and I'll give you a few seconds to decide. So you could see that the left image actually is the synthetically generated image. So I want to say that this is a cherry-picked example, and not all synthetic data is as realistic, but we can tune the synthetic data to be as realistic as we need, depending on how much realism we actually need. And in the next couple of slides, I want to show some use cases of synthetic data at various stages of medical imaging AI development and analysis. So DITSER has many staff fellows, ORISE fellows, and other researchers, and we have looked at a number of applications, starting from histopathology to breast imaging, and ending in kind of scorecard synthetic medical evaluation. And even beyond this, there are a number of other ways how synthetic data has been used in various for various applications in medical devices. So to start with, synthetic histopathology data generation. Nuclei and cell segmentation is a critical step in pathology image analysis. Quantifying cells can help improve the accuracy and efficiency of diagnostics and biomarker quantification. But developing AI algorithms requires accurate annotations of millions of nuclei and cells on a large whole slide images, which is often challenging. Imagine taking one of the images that is shown in the slide and then annotating every cell. In this project, my colleagues collected 4 histopathology datasets and developed and trained a diffusion-based generative AI model on pairs of histopathology images and their corresponding masks. They produce a synthetic histopathology image dataset that can be used for downstream task analysis. A pathologist confirmed that the results are very realistic and then can be identified stroma, carcinoma, and nuclei in the generated synthetic data images. The performance evaluation using synthetic data quality metrics shows high level of detail and a variety in the generated images. I encourage you to look at the RST and the links shown on the slides in this and other slides to learn more about each individual project. We also know that this is a different project that looks at pediatric AI usage and synthetic data. And we all know that kids are not just small adults, but in fact, their medical images vary substantially from adults. The anatomy, disease presentation, and image acquisition and quality are all different. All of these factors can contribute to variable performance in AI devices used to process or aid in interpreting these pediatric images. And AI devices are increasingly being used to optimize pediatric imaging, but collecting and annotating pediatric data, kids are a protected population, requires more expertise and thus is more challenging than adult data, meaning performance for pediatric patients in most AI devices is unknown. We have developed a synthetic pediatric imaging pipeline, as shown in the slide, that enables us to ensure that AI devices are safe and effective across a range of dimensions, where pediatric images differ from adults. The pipeline shown creates synthetic CT images using a virtual CT scanner, shown in the center. The resulting images are then fed into an AI model on the right. Since we know the ground truth patient and disease characteristics, we can evaluate the effects when comparing AI output to the ground truth. So this project is just one of many examples how synthetic data can give an opportunity to conduct virtual imaging trials to better understand the safety and effectiveness of AI devices in kids without unnecessary radiation exposure or expensive patient recruitment. In another project, we developed SSynth, a 3D knowledge-based and open source skin simulation framework for generating synthetic skin images with systematic variations of skin and lesion properties, as well as rendering. I will go into the detail of the pipeline in the second part of the presentation. Oops. In a follow-up project on NSynth, we also show that we can use synthetic training images generated by this pipeline to contain common skin artifacts to improve downstream performance on lesion segmentation tasks. So knowledge-based and simulation pipelines are not exclusive to skin. For example, in the NSynth project, where we focus on the creation of synthetic mammography datasets, containing 45,000 images. This dataset can be used to analyze AI model performance and find regions of parameter space for which performance varies from the aggregate level. Synthetic data can also be used to study disease progression over time. In this project, we introduced a computational model for simulating growth of breast cancer lesions, accounting for stiffness of surrounding anatomic tissues. A key aspect of synthetic data use is accurate reporting and evaluation. One of the tools developed by a group is the Synthetic Medical Data Evaluation Scorecard, which is a suggested comprehensive report to accompany artificially generated datasets. The scorecard is intended to help standardize evaluation and report performance according to a comprehensive set of metrics. I want to emphasize that imaging AI applications are only a part of a much larger set of applications of synthetic data use within medical devices. Computational modeling and simulation is an adjacent field. For instance, an algorithm in a device software may take in patient data and simulate the patient to obtain patient-specific results. I encourage you to look at the RST links shown below to learn more about this topic, which is beyond the scope of this presentation. So at this point, I've showed a number of applications, and you're probably wondering, how can I actually generate synthetic data? In this project, we created a kind of a taxonomy of different types of ways how synthetic data can be generated. We distinguished two different ways. Individual models, which are non-stochastic and intended to model an individual patient, so-called digital twin, or a family of patients that are related. On the other hand, population models are stochastic models that can be sampled from and create new individuals. Population models can be grouped into either image-based or knowledge-based methods. Image-based methods create synthetic data based on information obtained from patient images, such as CT scans. Generative AI is one type of imaging-based method that learns an AI model from images to generate new images. On the other hand, knowledge-based models take data from physical or biological measurements to create synthetic data. At this point, I want to pause and ask which of the following terms can be used to describe a digital representation of a patient's anatomy. I'll give you a few more seconds to respond. So the correct answer here is all of the above, which is C. So a phantom or a 3D model can be used to describe a digital representation of a patient's anatomy. So in this slide, I want to present an overview of different techniques that we created where synthetic data is used for radiology. And so I want to emphasize that there has been a lot of work done and across different fields, across almost every application in CT scans and X-rays, mammography, across both multiple modalities and anatomic sites. The type of models that are used, again, are varied. So many models are, again, imaging-based, generative adversarial networks, diffusion models, and so forth. But a lot of models are also simulation-based, patient-based, or knowledge-based, and so forth. And the colors in this table indicate are grouped by the type of model that's used to generate synthetic data, and whether the model is used to generate a particular patient or simulate, for example, an imaging acquisition device. I want to emphasize that a lot of the models today are imaging-based generative AI methods. And you probably see these models are very popular, and you may be wondering at this point, why would we want to use simulation? Why don't we just use the latest and greatest generative AI model that is often developed not for imaging data? So these are just some popular examples of models that can be used to generate images based on input prompts or other input data. So these models generate extremely nice outputs. But for medical imaging, one of the limitations is that there's a difference across the image space, for example, the space of where the X-ray sits, and the object space, which is the true representation of the object that you're trying to image, such as this patient. And so information is frequently lost during imaging operations. And so these two spaces are not bijective, and taking a particular point and mapping into the imaging space results in a loss of information. How does this manifest in practice? So in the middle, you see just a visual example, not medical, where you take images and then take out information from them and learn a GAN model on top of them. And you see that the resulting images may not be realistic. They may not be anatomically correct and contain artifacts. And so depending on how the synthetic data is used, it's important to be able to account for these kinds of limitations during the model. Another problem is that the outputs are often unconstrained, and again, depending on how the synthetic data generation model is parameterized, it may generate artifacts which may cause the resulting images to be unrealistic. So here's a very brief comparison of knowledge-based versus generative models. In knowledge-based models that obtain data not from images, but rather from physical or biological measurements, there are generally no null space limitations, while generative methods that are learning from images have a null space problem. As a result, generative methods may suffer from hallucinations, while knowledge-based methods may not. Knowledge-based methods are not a solution to everything. They may often be much slower to both implement and to run, and may often generate unrealistic outputs. On the other hand, generative methods learn directly from the distribution of images, and the outputs are often more realistic. However, you probably remember the example from earlier where I showed the breast imaging. In simulations, we can control the factors that we simulate and potentially can make very realistic data. And the choice of how much realism you need may depend on the type of application that you're studying. Another advantage of knowledge-based methods is control over biases, because we know what data is used to create a particular model. We have more control versus generative methods that learn directly from patient images, which are often suffer from limitations in patient data sets, propagate biases from the training data into the resulting synthetic data. So in summary, if you're going to choose a synthetic data generation technique, you may want to consider the quality and quantity of the source data that's used to create the synthetic data generator, whether there is available biological physical knowledge, as well as the application domain. Features such as the null space are important to consider, and object-based models decouple characteristics of the object from the imaging system. And again, the choice of techniques really depends on what is the risk of use of the synthetic data and how it is used. There's a term for this called context of use. And again, it's really important to be able to characterize what synthetic data is, how it has been developed, and how it is used, and what are the resulting risks. So in the next part of my presentation, I want to go back to the graph I shared earlier and look at some specific examples how knowledge-based models can be used to create synthetic data and in conjunction with AI development. In the first project, I want to discuss the MSIN dataset, which starts from a knowledge-based model of a breast developed by Graf and colleagues, which is then compressed. A mass is inserted into the breast, and then the resulting digital representation of a breast is imaged through a virtual simulator of a mammography device. This results in images where we vary characteristics of the breast and mass, such as the breast density, mass radius, and mass size, as well as characteristics of the imaging system, such as the relative dose with respect to the clinically recommended dose. We can see the effect of each of these variations of the images. You can see that in the images, for example, increasing the mass results in a mass that is larger size. The MSIM dataset is one of the regulatory science tools I mentioned earlier, and the full pipeline as well as the images are available open source via GitHub. So we look at some experimental validation of this dataset and look at one specific task, which is mass presence detection in digital mammogram images. This means that we take an image and output a single label with whether a mass is present in this image. For each specific breast density, we look at breast density and other properties. We generate a large data set and then compare it to a real patient data set, which in this case is 410 mammography images from the in-breast data set, which is a public mammo data set. And we use a metric called area under the curve, which is a standard metric for evaluating this type of task. So in this graph, we look at how does the training data affect the performance on mass radius and breast density. So here, each of the graphs shows the colors indicate the type of training data that is used to train a specific model, and the x-axis indicates the parameters of the testing data. And then the y-axis indicates the metric AUC, and in this case, the higher AUC is better. So synthetic data generates expected trends that are based on physics understanding of this task. We find that easier cases, for instance, masses with larger sizes or higher densities generated higher AUC values, including in-domain examples such as masses matching size or examples with matching breast density generated higher performance as compared to using out-of-domain data. Including all examples within a domain, for instance, all mass radii generates the best performance. So here I want to ask again another attention-paying question. What type of models suffer from null space limitations? Models trained from imaging data or models trained from physical and biological knowledge? Again, I'll give you a few more seconds to think about it. So correct answer here is A, models trained from imaging data. So in the next project, I wanted to discuss in more detail the SIM framework, which I mentioned earlier, which is an open source skin simulation framework for generating synthetic skin images, where we vary properties of the skin and lesions, as well as the properties of the rendering via detailed physics models to check how does AI downstream performance vary. So the framework for this is quite similar to the breast imaging that I discussed earlier, but the application is much different. And here I want to show a video of how this model was created. So we start with a model developed in Houdini, which is a multi-layered synthetic skin model that's procedurally generated. There's a blood network that is grown, as well as hair. and the lesion that is inserted into the skin. That each of the layers is assigned a material and rendered using virtual camera, using the Mitsuba framework, where we can vary systematically characteristics of all of the layers and the imaging to generate the resulting images. So some of the variations that this model allows are shown here. The melanosome fraction, which is a proxy for skin color, the variation of which is shown on the left, shows that we can generate for the same exact model of a lesion in skin, we can generate variations in skin tone. And we can also do this for other parameters such as the blood fraction, and vary, again, these characteristics based on values that we obtain from literature. We can also look at binary characteristics. For example, we can add and subtract hair from the same exact model. We can also make the lesion shape more regular or more irregular, depending on what we want to study. So this model allows us to look at, given a particular skin segmentation model that's trained on only patient data, how does the performance vary with these annotations? I want to emphasize that for skin imaging, there are the patient data sets are extremely limited, and virtually none of the labels that are seen in this slide are available for patient images. And so we can actually test the variations that are not labeled in patient images using these models. We find that synthetic test images with hair artifacts resulted in lower performance compared to the images without hair artifacts. The performance drops with darker skin color, which is a known issue with many skin imaging AI tasks. It is slightly easier to segment lesions that are more regularly shaped than irregularly shaped. And also the blood fraction slightly affects performance. We can also add synthetic data during training and see what happens. So in this graph, in set of graphs, I look at how does the proportion of patient to synthetic data affect performance. And so the x-axis indicates the proportion of patient data to synthetic data, while the y-axis indicates the DICE score, which is a segmentation metric, and the higher DICE, the better. And so the left graph shows performance when the model is trained only on real data and then evaluated on a held out test data set. So test data sets are the same. It's this patient data set used in all three graphs. The dashed in graphs are the HAM data set, and the kind of filled in graphs are the ISIC data set. The difference between these two data sets is ISIC is much smaller than HAM. And so you can see that if we only had real patient data, which is the graph on the left, we only use 20% of the data of each data set. The performance is much worse than if we had 100% of the data. So this shows the more real patient data we have, the better. If we kind of consider a simulated scenario where we replace portions of real patient data with synthetic data, which is the graph in the middle, we find that if the model is trained only on synthetic data, the performance is not that great. But if even 10 to 20% of the patient data is replaced by, if 10 to 20% of the real data is used and then the rest is synthetic, the performance is actually nearly the same as if the entire patient data was used. And finally, if we add different subsets, if we add synthetic data to the full real data set, which as you can see on the right, the performance also improves slightly. So we find that addition of synthetic data sets to small patient data sets generally results in improved performance for this task. So this concludes the ESSENT project. There are a number, however, of remaining challenges and considerations that are important to look at with the use of synthetic data for these applications and others. So there are a few remaining challenges I want to highlight. How do we evaluate synthetic data sets? What are the appropriate metrics, and how do we actually sort of check if they can or cannot be used in a particular context? many of the methods that we see today on synthetic data are imaging-based. And how do we address the limitations of the imaging-based models? Do we need to address them? And how do we address dependency, for example, on the acquisition device that was used to collect a particular data set, and then we wanted to generalize? In the context of knowledge-based and simulation-based methods, how can we simulate more realistic knowledge-based data for it to be able to use it in a practical context? So responsible use of synthetic data in AI development has promise in resolving gaps in real patient availability and variability. But not all synthetic data is alike, and it's important to consider, again, the context of use and the risk associated with a particular application where synthetic data would be used. And we have developed and disseminated several regulatory science tools and publications that contain knowledge-based pipelines as a proof of principle for developers for using these synthetic models. And I encourage you to look at the regulatory science tool catalog to learn more about them. So this concludes my talk. Please feel free to send me an email for the slides. And I think it would be a good time to open up for questions. Thank you.
Yes, thank you. So far, we have 2 questions. Are there any more questions? You could place them in the Q&A. I did get some requests for the slides. My department cannot share the slides, and we will not be able to post them. But like Elena said, if you want to email her for the slides, that will be fine. First question: why knowledge-based methods may produce unrealistic results, while generative methods may produce more realistic results?
So this is a great question. If you look at the synthetic images here, for example, this is a skin model. You could, again, it's hard for me to tell because I look at these images all the time, but they generally don't look like a realistic image of skin, right? To some extent, depending on the resolution. But it's important to look at all of to make a simulate a realistic image. It's important to look at characteristics such as light, reflection, the underlying kind of parameters, and tuning these parameters is very hard. Versus if you start with just patient images and learn kind of a statistical generative model on top of them, it is easier to learn that kind of distribution because you start from a set of patient data, imaging data, and then generate the result.
Thank you. Next question. You mentioned that knowledge-based methods allow better control over biases as compared to generative methods that usually incorporate biases from the training data. Could you elaborate more on that? Additionally, which part of the pipeline does this fit into?
So again, I would give an example from the SSynth framework. So I kind of glanced over this, but here is a distribution of one of the data sets of skin colors of one of the public data sets that we use, as well as the synthetic data distribution that we generated. So you could see that both of these distributions are not sort of equal or not equal by skin tone. But specifically, if you're learning, let's say, a diffusion model of the skin images that are distributed based on the skin tone shown on the real data distribution on the left, then you're probably going to get this type of distribution as an output. Versus in knowledge-based models, we can choose to manipulate the melanosome fraction as well as the imaging to control this distribution, which would be much more difficult to do if you already have a fixed set of images where you only have very few dark skin patients.
Thank you. Next question. How does adding or replacing real data with synthetic data differ from using augmentation methods?
So this is a great question. So you can think of synthetic data as a form of data augmentation. kind of a modified form of data augmentation. And so in the experiment, for example, with addition or replacement, again, we want to systematically vary proportions of synthetic data and patient data subsets. So I think one key difference is how, again, how the synthetic data is generated. So for example, a common data augmentation method is that is not synthetic data based. You're flipping the images and then using sort of the resulting flip images in addition to the original ones to improve your performance. But you're really using the same distribution in terms of, for example, skin tones. That doesn't really change. And so if you're also maybe augmenting data that's comes from a GAN or another generative model, you're also using a very similar distribution of data as in addition to the original training patient data. Versus if you're using kind of simulations or external information, you're adding other distributions to try to balance performance, either overall or balance or improve performance, either overall or a particular subgroup.
Great, thank you. Could we use generative method to create data set for performance evaluation for an FDA submission?
So that's a great question. I wouldn't be qualified to answer regulatory questions on the webinar, but there are submissions sort of we see at FDA that use synthetic data, and it's an emerging trend. Oftentimes that is used for training. And again, one of the important things to consider here is how do you sort of, if you're using synthetic data, is this slide. So what information do you have for making the synthetic data and how it is created? So it's not that different from if you're just using AI, you're just using a different type of AI to make data. Or if you're using a simulation framework, again, what is it that you're doing and how it is used and what are sort of associated risks and contexts of uses.
Great, thank you. Are there any regulations from the FDA with regards to approvals when synthetic data is used to train the models?
So again, this is a great question. At the moment, I believe we do not have a guidance in terms of specifically for synthetic data, but I would also encourage you to look at the CMNS guidance that I mentioned earlier to learn more about sort of current stage.
All right. The next question has a couple of parts. I'm going to read them all. Great talk. Thank you. Mixing synthetic and real data sounds like the least burdensome approach. How do you validate synthetic data generation? Is it a mostly quantitatively driven assessment like the breast image given earlier?
So again, as we see more synthetic data use, it's important to consider sort of, again, how it is used and in what context. And so we are learning this as the industry expands and then there are more applications. So again, it's important to look at, so the scorecard project is kind of an initial effort to try to come up with a kind of a standardized evaluation and reporting for synthetic data. So depending on the type of application, it may be important to look at either task-based assessment or sort of non-task-based assessment. So yeah, I think the answer here, it depends on your task.
All right, thank you. Next question. To date, has FDA given marketing authorization to any devices that included synthetic data in training?
I won't be able to answer that, sorry.
This is more of a note. They want to see your email address again.
Sure.
You could put the slide with your email address. All right, next question. These are all They're rolling in, and I think we have 10 more minutes to go. So are you fine, Elena?
Absolutely, yep.
Great.
Ask away.
Okay, great. For the synthetic images generated in downstream analysis, were you just experimenting with different proportions of synthetic plus real images and how it affects the performance? For example, adding 50% of synthetic data improved performance on the downstream task.
So we did experiment again for so the slides have a very kind of brief overview, but we did experiment. For example, there's a the closest here is 30 to 70, 40 to 60, 60 to 40. But here, these kind of splits are a proportion of like half real patient and half synthetic data sets. And so what happens to the dice performance? So that's what we were doing. And I think the replacement experiment is actually a very nice way to look at sort of, we have the full data set available for each case here. And so in a hypothetical scenario, if we didn't have that case, for example, and then sort of a new use case or something else, what would happen if, you know, we actually use synthetic data to supplement the limited patient data, but we have sort of a reference standard to compare, sort of an oracle to compare, that if we had the full data set, this would be the final performance.
Are these data sets specific for some spectrum of imaging equipment? E.g., would an image obtained on 0.72 and 1.52 MRI give you similar results after training and validation?
So we haven't really considered MRI simulation in sort of these projects, and one of the reasons is that it's challenging to be able to simulate a sort of a very specific MRI machine in comparison to, for example, a mammography image, where in mammography we used very specific detectors that were kind of indicative of a particular manufacturer, just as an example. But again, it sort of, depending on the application, you really want to think whether it's mammography, whether it's MRI, whether it's skin, you really want to, you probably know best what are the differences across the detectors or imaging devices. And so, and how these differences would affect the downstream performance.
Thank you. Is there a correlation of model AUC and the higher spectral resolution of the data set? Does this apply only to patient-derived versus synthetic sources?
So if you generate synthetic images, so absolutely resolution matters, right? We can downsample these images to, you know, very few pixels kind of resolution size, and then we won't be really able to see the masses. So we haven't explicitly conducted an experiment where we compare kind of the effect of resolution, but actually it would be an interesting experiment to conduct. And the other thing I wanted to note is that we are able to simulate images in both masses, in both the mammography project and the skin project, at extremely high resolution. And then for the downstream AI performance, we actually downsample them to a smaller size because we're constrained by the architecture. But absolutely, the resolution is important, and simulation actually allows you and us to compare some of these effects, versus if you are looking, for example, at a data-driven model, it may be constrained to the resolution of the input image. And so it would be more difficult to generate images at a higher resolution without sort of appropriate super resolution techniques.
Great, thank you. This next one, I'm not really sure if you can answer it, but I'm going to ask it anyways. Will FDA accept a submission package of AI as medical device where a portion of the data used for AI training is synthetic.
So again, this is a kind of a more of a overview talk. So the FDA has a mechanism called regulatory mechanism called USAB, and so any kind of questions that would be for a particular device, that would be a great way to sort of interact with the FDA and to ask these type of questions. But I do want to emphasize that synthetic data, again, is not used in every submission, but certainly we do see submissions with synthetic data.
Thank you. Thank you. Are there models that combine both knowledge-based methods and generative methods?
That is also a great question. So there are models, oftentimes they're called hybrid or physics informed. So again, there are many different names for these models. So I would encourage you, so specifically for radiology, you can look at this survey of techniques. So you can see the title on the top right. But hybrid models actually allow sort of, in many cases, to take the best of both worlds. I hope there are more models developed in this domain.
Thank you. Are there models that combine both knowledge-based methods and generative methods?
Yes, so that would be hybrid or physics informed models. Maybe there are other names for them.
Is the evaluation of synthetic data different from validation of synthetic data?
I think this is a terminology question. So I think a key distinction would be, if the synthetic data is generated, evaluated on itself or in comparison to the patient data.
Thank you. Could you elaborate more on the concept of null space?
So the null space would be the concept shown here. So you start with an imaging space, which is, for example, a lossy space. Again, here you just see an example, but imagine sort of a set of information that is lost, such as you lost certain sort of piece of the sort of the head here or anything. But essentially you start with a breast or another kind of object that is kind of from a patient, right? Then you image it. And so this results in a loss of information. And so by learning on the lossy measurements, you're not necessarily learning to generate the full model, and which may cause kind of various downstream issues. And in addition, if your model is under constraint, there are sort of both null space and hallucination issues that would occur. And so you would want to, again, depending on how much they actually affect your performance, you may want to be able to account for them.
Thank you. We're getting kind of short, so I don't know. I think we'll maybe do two more questions. For the breast example, if an AI detection algorithm is trained only on real cases and tested on synthetic data, does it give the same result as if it was tested on real cases? Assuming the test and synthetic data sets are matched.
So this is also a great question, really getting at this project. Unfortunately, I don't have the graph of this. I only have the synthetic results. But in practice, we found that no, for the breast project at the moment, this is not the case. So if you look at the breast images here, there are sort of windowing and other important processes that need to be need to be added to the images to make them more realistic. So when we kind of qualitatively compared the synthetic mammography images to the in-breast and other patient images, they're clearly distinct. But the MSynth project sort of was just a demonstration to show that we can, in fact, use this pipeline and then obtain performance trends that are similar to what we would have expected in patient data. But there sort of more work needs to be done in terms of actually improving this whole pipeline to make it more realistic. Whether it's post-processing the images to make them more realistic, or whether it's adding things like calcifications to the images, maybe to add more realistic confounders, or other things that would sort of adjust the synthetic to real gap.
Thank you.
And synthetic to real gap is kind of a big problem in many of these methods, but again, with enough work, it may be adjusted.
All right, we got a minute left. I'm going to ask the very last question here. Thank you so much. As a rule of thumb, I've heard from the FDA is 50% of test data for AI/ML enabled devices must come from the US. Is there a similar rule of thumb for synthetic to real data ratio?
Again, this is more of a regulatory question. I think, again, take a look at the Q-sub mechanism and other sort of official question mechanisms.
Great, thank you. Thank you. Thank you all for so many great questions. They're still rolling in, and I think we got only to about half of them. But thank you so very much for your presentation and presenting your research and data, Dr. Skizakova. Really appreciate it, and good luck to your future research. Very, very informative Q&A session. Thank you, everybody, for attending our grand rounds. That's it for today. Thank you.
Thank you so much.
Recording stopped.